Mr. Maxwell for Pingree AP Biology. I want to talk to you today about some essential things you need to know about membranes before you can do the water potential problem set. So, here's what you should know. You have to know, here's your basic phospholipid bilayer, which is the heart of a membrane. You should already know that there is a nonpolar portion that is composed of the fatty acid tails. And you should also know that the phospholipid heads are polar and therefore hydrophilic. So hydrophobic center, hydrophilic outside. And if you take a look at something like carbon tetrachloride, which is nonpolar, you should be able to predict that carbon tetrachloride will flow through the membrane pretty easily because like dissolves like, and nonpolar things are perfectly soluble here in the hydrophobic tails. Now, you might question what it's doing out here in the polar environment or what it's doing here in the polar interior, but that's another story that we're not going to talk about at this moment. We should also look at water, and by the way, you should notice that these things are not drawn to scale. The water molecule here is way bigger than it should be, but this is for illustrative purposes. Water molecule, of course, is polar, and therefore it will happily interact with the polar heads here, but it will not interact with the fatty acid tails to any real appreciable extent. Now, in older textbooks, you will see water molecules squeezing their way through this fatty acid tails, but the work of Dr. Peter Agre, who won the Nobel Prize for a discovery of aquaporins, shows that that doesn't happen to any real appreciable extent. So, of course, it happens a little bit maybe, but not to any appreciable extent. And then we also have things like sodium, which is charged, and therefore it's going to interact with polar things. It's hydrophilic, and not with the nonpolar interior. Or, of course, likewise, negatively charged particles also are going to be perfectly able to dissolve and interact with polar things, but will tend to be excluded from nonpolar hydrophobic region of the membrane. So this is an aquaporin, and this is how water gets through membranes. Aquaporin is a really cool molecule because it has a channel, of course, it's hydrophilic, and actually has the correct shape and charge distribution to allow water, and pretty much only water, get through it. There are individual channel proteins for sodium, for chloride, for potassium, and each of them has the particular shape that it needs to have and charge distribution that it needs to have to allow that ion and only that ion to go through. So this allows different cells to control what they're permeable to by expressing different proteins in those membranes. So why do things cross membranes anyways? Well, the simplest form of transport across membranes is simple diffusion. And osmosis is a special form of diffusion across a membrane, and we'll talk about that too, but suffice it to say that things tend to move to where they're not. And so here we see a molecule moving from an area of high concentration to an area where it's not very concentrated. And that'll happen until there's a balance where particles move across, particles move back, and here they're moving at a fairly rapid rate. Here there's some moving back, but they're going to move mostly down their concentration gradient. Remember, there's always chaos in the system, and so things will move back and forth, but the net flow will be down the concentration gradient. And it's important to remember that molecules do not have brains, and therefore they do not want to move down their concentration gradient. They simply do because they move and bounce off of other things, and if they move and bounce off of other things, when they bounce, they will be moving in a direction which there are less things on average. And so that's really the essentials of diffusion is moving and bouncing in a direction in which there are fewer things. Not on purpose, not because you have a brain, not because you want to, just because there's less stuff to collide with once you've collided with something. Okay, so we can refine this idea a little bit, and here we have a membrane, and we have some solute molecules, which in this case can't move across the membrane. However, we're going to say that water molecules can move across this membrane. And so water, since the solute can't move, the water is going to need to move such that the concentration of water on both sides of the membrane becomes equal. So on the left we have a dilute solution, on the right side we have a concentrated solution, and water is going to move to make the concentrated solution more dilute. Not because it has a brain, but because a simple matter of bouncing off things. So water is going to move down its concentration gradient. We say that this right side here is hypertonic, and this is a hypotonic environment, and water will move into a hypertonic environment from hypotonic environment. So on the left side, low concentration of solute, right side, high concentration of solute. Because there's a high concentration of a solute on the right side here, there is a low concentration of water relative to the amount of solute, and therefore a high concentration of water here on the left side relative to the solute. And so water is going to move in a net water movement. It's going to be to the right here. If you have more solute molecules, it's going to be more hypertonic. And so water potential is a concept that allows us to quantify exactly how hypertonic or hypotonic something is. The problem set deals specifically with water potential. Water will always move from an area of high water potential to an area of low water potential. Water potential is a concept that's often used in plant systems, very often used in plant systems. You'll see in animal systems, more often they'll use the concept of osmotic pressure, although these things are essentially equivalent concepts. Water moves from where there's low osmotic pressure to where there's high osmotic pressure. And so we talked about hypertonic, isotonic, and hypotonic, and you should know these terms, hopefully from freshman bio, but in a hypertonic environment, the cells themselves will have to be hypotonic, and therefore the net flow of water will be out of the cell, and the cells will shrink. In a hypotonic environment, on the other hand, cells will be hypertonic, and therefore the net flow of water will be into the cell. And this can be unhappy for a red blood cell or general animal cells because they'll swell up too much if they're too hypertonic to the environment. Typical environment is for animal cells is the isotonic to their environment, and there is flow of water in and out of the cell, but there's no net flow one way or the other. So that's an isotonic environment. The cell would therefore be isotonic as well. For most other types of cells, cell walls are a really common thing. Animals are kind of the exception to the rule. Plants, fungi, and most bacteria have cell walls, and so this is a more common situation in life. And here we have a hypertonic environment where the water is getting sucked out of the cell because the cell is hypotonic, and therefore the cell has no choice but to become smaller. Right? And as it becomes smaller, it will plasmolyze, and that means the membrane will pull against the cell, away from the cell wall. Whereas in a cell that has a lot of solute inside it, it will be hypertonic to the hypotonic environment. Net flow of water will be in, and there will then be pressure against the cell wall, making it rigid, as opposed to an environment where the cell is isotonic, and there's no net flow of water either way. The pressure against the cell wall goes to zero. And this is actually not a healthy situation for plant cells because plants need that turgor pressure to be able to hold themselves upright. And finally, since we introduced the concept of more and more and more hypertonic environments, water will flow from where it's a high water potential to where it's a low water potential to where it's an even lower water potential. So it's really difficult for some people to think of 500 as a smaller number than 0, but the little sign here makes it negative, and therefore a low water potential, negative 500 kilopascals, is a low water potential relative to negative 200, and negative 200 is low relative to 0. And so water is going to flow down its concentration gradient to where it's less concentrated. It's going to flow down its water potential into an area where there's a lower water potential. So that's the essential stuff you need to know in order to get to work on the problem set. So you can go ahead and download that and get to work on it. Thank you.